Now let's start with the historical background of photosynthesis. In this historical background, I am going to explain about the experiments performed by different scientists to identify the key features of photosynthesis. Okay, first of all, let's start with the experiments of Joseph Priestley. Experiments conducted by Joseph Priestley. Okay, so Joseph Priestley performed a series of experiments by using bell jar. Hence, these experiments are also called as bell jar experiments. So, in his first experiment, he has taken a bell jar, closed bell jar, and in this bell jar, he placed a burning candle. And after some time, he observed that this burning candle got extinguished. It has got extinguished. Okay, next, in his second experiment, he performed second experiment. In his second experiment also, he has taken the same type of bell jar, which is closed bell jar, inside which he placed a burning candle again. Along with this burning candle, now he placed a living mouse. Now I am representing this mouse with M, living mouse. After some time, when he observed this bell jar, he observed that the candle got extinguished again. It got extinguished again, turned off, and mouse was dead. Next, he performed third experiment. In his third experiment also, he has taken the same type of bell jar, and inside this bell jar, again he placed burning candle. After some time, burning candle, and again he placed mouse. Along with mouse, he also placed a potted plant. That is mint plant. After some time, when he observed this bell jar, he observed that candle continued to burn. That means it was still burning. Candle was burning and mouse was alive. Okay, now let's see what is the reason for the burning of this candle and for mouse to be alive. Next, so here what is happening, due to the burning of candle and due to the respiratory activity performed by mouse, foul air is produced or dirty air, which is called phlogiston. So due to burning of candle and the respiration performed by mouse has released dirty air, which is foul air, which is also called as phlogiston. Okay, now the mint plant, which is placed inside this bell jar, purified this phlogiston, that is foul air. Plant has produced air which purified the phlogiston. Now this purified air is called as, is nothing but oxygen. This purified air is called as dephlogiston because it is purifying phlogiston. Dephlogiston. Okay, right. Now this phlogiston is nothing but carbon dioxide, which is released by the burning candle and by the respiratory activity of the mouse. Now this dephlogiston is nothing but oxygen. Okay, so why this plant is purifying this phlogiston? Let's see. So actually what happens, due to the burning of candle and due to the respiratory activity of this mouse, what will be produced? Carbon dioxide will be produced, and this carbon dioxide is absorbed by plant for the process of photosynthesis. So by the end of photosynthesis, plant produces oxygen. Now this oxygen which is produced by plant by the process of photosynthesis is absorbed by candle, and it is also absorbed by the mouse to be living, and hence mouse was under living condition and even candle was under burning condition. So this is all about the experiments performed by Joseph Priestley, and finally he said that whatever is removed from the atmosphere, whatever is removed by burning candle and the living mouse from the atmosphere has been restored into atmosphere by the plant, by the green plant. What is that? Oxygen. So oxygen is removed by burning candle and even by the living mouse to be under living condition, and that oxygen which is removed from the atmosphere has been restored to atmosphere by this plant through the activity of photosynthesis. Okay, so this is all about the experiments performed by Joseph Priestley, and finally he discovered oxygen in the year 1774. 1774. Okay, next let's start with the experiments performed by Jan Ingenhousz. Now let's study the experiments performed by Jan Ingenhousz. Okay, Jan Ingenhousz, he used the similar setup which was used by Joseph Priestley. He used the similar setup which was used by Joseph Priestley, and he placed this setup once in dark and then once in light. Then what he observed that when he placed this setup under light, when he exposed this setup to sunlight, he observed that there is purification of air by plants. So purification or production of purified air. Pure air is formed. Purified air is formed by green plants when this setup is exposed to light. But when this setup is exposed to dark, this purification of air is not performed by, or purified air is not formed by the green plants. That means here he showed that, he said that light is essential for the process which is required for the purification of air. So light, sunlight is essential for the process to occur which is required for the process, for the purification of air. Okay, he also, along with this experiment, he also performed another experiment in water plants, that is aquatic plants. So in this experiment, he has taken a beaker. In this beaker, so this is a beaker. In this beaker, he has taken an aquatic plant, that is hydrilla. So this is an aquatic plant called hydrilla, and this plant is covered by an inverted funnel. This plant is, he covered this plant with an inverted funnel. Okay, and then he illuminated this setup with light, or he exposed this setup to sunlight. So after some time, he observed that there is formation of bubbles. There is release of bubbles from green parts of the plant. Later, these bubbles were tested and found that these bubbles are with oxygen. So these bubbles are with oxygen. When these bubbles are formed only when the setup is exposed to light. When the same setup is exposed to dark, there is no formation of bubbles. He didn't observe the formation of bubbles. That means in the presence of light only, and that too from the green parts of the plant, these bubbles are formed. These bubbles were tested later, and these bubbles are with oxygen gas. Oxygen gas. Oxygen is nothing but, this oxygen is purified air which is purifying, and this process is nothing but photosynthesis. The process which is involved in the production of this oxygen or purified air by green parts of the plant in the presence of sunlight is photosynthesis. Now we are studying that process as photosynthesis. Okay, after Jan Ingenhousz, Julius von Sachs, another scientist, Julius von Sachs performed a series of experiments on green plants. Julius von Sachs performed a series of experiments on green plants, and he stated that green parts of the plant are able to synthesize food in the form of glucose, and that glucose is stored in green parts of the plant in the form of starch. That means glucose, after its synthesis through a process called photosynthesis, which we are studying now, that process as photosynthesis, is polymerized into starch, and after its polymerization into starch, it is stored inside the green parts of the plant. Okay, so by now, that is by the mid of 19th century, scientists have come to know the key elements, important elements which are required for the process of photosynthesis, and hence by the mid of 19th century they have given the empirical equation of photosynthesis. Now let's study the empirical equation of photosynthesis. So now the empirical equation of photosynthesis, which is given by the mid of 19th century. Empirical equation of photosynthesis. Okay, so what happens during the process of photosynthesis? Carbon dioxide and water results in the formation of carbohydrate, that is glucose, and oxygen is also released in the presence of sunlight and chlorophyll, right? So according to the earlier view of scientists, the oxygen which is released during the process of photosynthesis is coming from carbon dioxide. But now we are studying, we know that this oxygen which is which is formed during the process of photosynthesis is coming from water but not from carbon dioxide. So photosynthetic oxygen is contributed by water but not by carbon dioxide. So to prove that this oxygen, photosynthetic oxygen, is coming from water but not from carbon dioxide, 3 scientists performed 3 different experiments to prove that this oxygen, photosynthetic oxygen, is coming from water but not from carbon dioxide. Those experiments are Van Neil's experiments. Van Neil is a scientist. He performed his experiments in bacteria, that is purple sulfur bacteria and green sulfur bacteria. Purple sulfur and green sulfur bacteria, and proved that oxygen is coming from water but not from carbon dioxide. And these experiments have become the milestone contribution to the understanding of photosynthesis. Okay, next experiments are performed by Robert Hill. So Robert Hill also performed experiments to prove that this oxygen is coming from water but not from carbon dioxide. He used Stellaria. It is an angiospermic plant. He used an angiospermic plant called Stellaria as an experimental material. Next, after Robert Hill, 2 other scientists, Ruben and Kamen. These 2 scientists together, they also performed experiments in a unicellular blue-green alga, that is chlorella, and proved that oxygen is coming from water but not from carbon dioxide. Now let's see the experiments of Van Neil, what he stated with his experiments. Okay, now let's study the experiments of Van Neil, which he performed on photosynthetic bacteria like purple sulfur bacteria and green sulfur bacteria. So in green sulfur bacteria and purple sulfur bacteria, instead of water, they use sulfurous compounds instead of water. Actually, during plant photosynthesis, carbon dioxide and water is utilized for the process of photosynthesis for the formation of carbohydrate. But here, in case of green sulfur bacteria and purple sulfur bacteria, instead of water, sulfurous compounds are used, due to which carbohydrate is formed. Along with carbohydrate, what is released? Sulfur is released. Actually, in plant photosynthesis, what will be released as oxidation product? Oxygen is released as oxidation product. Why oxygen is released as oxidation product? We studied in the previous video. I just repeat it. So, in case of plant photosynthesis, what happens? 6 molecules of carbon dioxide plus 12 molecules of water is consumed. And in the presence of sunlight and chlorophyll, it results in the formation of C6H12O6 plus 6 molecules of water plus 6 molecules of oxygen is released. So here oxygen is released. Oxygen is released because water undergoes oxidation into protons, electrons and oxygen. So these protons and electrons are picked up by carbon dioxide, so that carbon dioxide is reduced into carbohydrate here, and hence the oxidation product is oxygen. So due to which oxygen is released as oxidation product through the process of photosynthesis in case of plants. Now let's study in case of green sulfur and purple sulfur bacteria. So when he conducted experiments on this green sulfur and purple sulfur bacteria, he observed that there is release of sulfur instead of the release of oxygen. So why this sulfur is released? Because these green sulfur and purple sulfur bacteria are utilizing sulfurous compounds instead of water. So here this sulfurous compound is H2S, hydrogen sulfide. So this H2S undergoes oxidation into protons, electrons and sulfur. So now this sulfur is released as oxidation product, and these protons and electrons are picked up by carbon dioxide, which is reduced into carbohydrate. And the type of photosynthesis in which there is no release of oxygen is called as anoxygenic photosynthesis. So the type of photosynthesis in which there is no release of oxygen is called as anoxygenic photosynthesis, and the type of photosynthesis during which oxygen is released is called as oxygenic photosynthesis. So oxygenic photosynthesis is performed by green plants. Oxygenic photosynthesis is performed by green plants, whereas anoxygenic photosynthesis is performed by bacteria, and some bacteria like cyanobacteria. So which type of bacteria perform anoxygenic photosynthesis? Green sulfur and purple sulfur bacteria. So green sulfur and purple sulfur bacteria perform anoxygenic photosynthesis, but a type of bacteria, that is cyanobacteria, perform oxygenic photosynthesis because they also utilize water during the process of photosynthesis. Hence the oxidation product or the metabolic byproduct will be oxygen. So cyanobacteria perform oxygenic photosynthesis. Okay, so this is all about the experiments performed by Van Neil. So after performing these experiments on green sulfur and purple sulfur bacteria, Van Neil stated that photosynthesis is essentially a light dependent process during which protons or hydrogens from a suitable oxidizable compound, from a suitable oxidizable compound are involved in reducing carbon dioxide into carbohydrate. So here what we are studying, from a suitable oxidizable compound during the process of photosynthesis, hydrogens or protons from a suitable oxidizable compound are reducing carbon dioxide into carbohydrate, even in case of bacteria also and even in case of plants also. Here from a suitable oxidizable compound, that is water, protons are coming and reducing carbon dioxide into carbohydrate, whereas in case of purple sulfur and green sulfur bacteria, from a suitable oxidizable compound, that is H2S, which is undergoing oxidation, protons are coming and they are reducing this carbon dioxide into carbohydrate. So this Van Neil's experiments are the milestone contribution to the understanding of photosynthesis. Now let's start with the experiments of Robert Hill. Okay, now let's study the experiments performed by Robert Hill. So Robert Hill performed his experiments in an angiospermic plant, that is Stellaria. So to perform his experiment, he has taken a beaker. In this beaker, he filled this beaker with water, and in this water he placed the chloroplasts of Stellaria plant. So these are the chloroplasts of Stellaria plant. Chloroplast of Stellaria plant. Okay, right. So after taking chloroplasts of Stellaria plant, he exposed this setup to sunlight. After exposure of this setup to sunlight, he observed the formation of release of bubbles in this medium or in this water. And later, he tested these bubbles. He tested these bubbles and he found that these bubbles are with oxygen gas. Oxygen gas. So now what can we say? This oxygen is releasing from water but not carbon dioxide, because in this water, in this experiment, that means in this suspension only water is present but there is no carbon dioxide. Carbon dioxide is absent and hence obviously this oxygen is releasing from water. How? Because this water undergoes splitting due to sunlight. So when this experiment setup is exposed to sunlight, this sunlight is involved in splitting of water. So this water splits into protons, electrons and oxygen. Protons, electrons and oxygen. So what happens? This oxygen is released as the oxidation product. So here I would like to discuss one more point here. He also, to confirm that this oxygen is coming from water but not from carbon dioxide, he added other type of chemicals like benzoquinones. To this suspension he added benzoquinones, either benzoquinones or ferricyanides or chromates, or he added DCPIP. So any one of these chemicals can be added to this suspension to prove that this oxygen is coming from water but not from carbon dioxide. So now let's talk about DCPIP. DCPIP is nothing but dichlorophenol indophenol. This dichlorophenol indophenol exists in two forms, that is in oxidized state and reduced state. In its oxidized state it is in blue color, whereas in its reduced state it is colorless. Okay. So when this oxidized state, he added oxidized state of DCPIP to this suspension. Oxidized state of DCPIP to the suspension. So what happens? This entire medium this suspension becomes blue colored. The suspension is becoming blue colored. Okay. So after addition of this DCPIP, oxidized state of DCPIP to this suspension, then he again exposed this setup to sunlight. So when he exposed this setup to sunlight and observed after some time, what happened? Again bubbles are released. These bubbles, later he tested these bubbles and found that these bubbles are with oxygen. So what is happening here? After formation of, when the bubbles are formed, he observed that this suspension has become colorless. Why this blue colored suspension has become colorless? Because water present in this suspension undergoes splitting, due to which protons and electrons are released. Along with that, oxygen is released in the form of bubbles. These protons and electrons which are released inside the water due to splitting of water are transferred to oxidized state of DCPIP present inside the water, and hence this DCPIP will be converted into its reduced state, and hence water becomes colorless. So once the water becomes colorless, we can say that these protons and electrons which are released from water are transferred onto the oxidized state of DCPIP, which was in blue color, and hence this oxidized state will be converted into its reduced state, and hence it becomes colorless, and hence entire suspension becomes colorless. So in this manner he proved that oxygen is coming from water but not from carbon dioxide during the process of photosynthesis. Okay, let's study next experiments. Last experiment performed by Ruben and Kamen. So Ruben and Kamen performed experiments on chlorella, which is unicellular green alga. So they performed experiments on chlorella, which is unicellular green alga. So in their experiments, they have taken a beaker, and in this beaker they have taken some water, and inside this water they placed this chlorella, that is unicellular green alga, chlorella, unicellular green alga, chlorella. Okay, right. So now the water which is taken in this beaker is with radioactive isotope. this water is with oxygen. The oxygen present in this water is with radioactive isotope, that is O18, radioactive, and it is heavy. And the carbon dioxide which is provided in this suspension is with normal oxygen, that is O16. So and then this setup is exposed to sunlight. When this setup is exposed to sunlight, after some time, when he observed this setup after exposure to sunlight, after 1 to 2 hours of exposure to sunlight, then he observed the release of oxygen gas. He observed the release of bubbles and he tested these bubbles, and these bubbles are with oxygen, and now this oxygen is with O18 isotope. O18 isotope. That means this oxygen is released from water but not from carbon dioxide, because carbon dioxide, the oxygen present in carbon dioxide is with normal oxygen, that is O16, whereas oxygen present in water is only the radioactive isotope, that is O18. And the oxygen which is released through the process of photosynthesis is also with radioactive isotope, that is O18, and hence it is proved clearly that oxygen is coming from water but not from carbon dioxide, with all these 3 experiments performed by Van Neil, Robert Hill, and then Ruben and Kamen. So this is all about the experiments performed by different scientists to identify some key features of photosynthesis. In the next video, I will be starting with the site of photosynthesis. Thank you, students.